We so often think of evolutionary change as an isolated process, specific to a species who gains an adaptive advantage, experiences stochastic drift, or even just undergoes a demographic change, a bottleneck, for example. But what do we know of the effect of evolutionarily adaptive measures on organisms' ecological systems? Could an adaptation, or as we will see, a maladaptation, of one organism pose selective pressures on that species' surrounding varied populations?
Now, we won't pretend as though there's one answer, or that the process itself can be definitive, but we're going to take a closer look at one case study by Patrick Nosil's lab in the UK. Nosil's lab takes a particular interest in Crypsis and Timema Cristinae, or stick bugs, these little guys. Want to find out more? Stick around and we'll bug you with the facts.
Crypsis is the ability of an animal to avoid detection by other animals, and no doubt you've seen this in numerous examples. Chameleons, octopi, butterflies even. But more than just increasing the fitness of the individual or a particular species, Crypsis has an important role in evolutionary dynamics. But of course, we usually think of Crypsis as visual camouflage and mimicry. Talk about strong evolutionary pressures. Can you find the bug in this picture? What about here? Not so easy, is it? These are Timema Cristinae, stick bugs. Nosil's lab takes a particular interest in how the evolution of camouflage in Timema Cristinae drives rapid ecological change, changing the composition of the insect communities that live in Simpatry.
Stick bugs are abundant on two different shrub species, and for this reason, have two dominating ecotypes: striped to suit adenostoma host plants, or green to suit ceanothus host plants. However, due to gene flow between populations that results in imperfect camouflage or maladaptation, the wrong ecotype match leads to varying effects.
So Nosil's lab basically found that populations of stick bugs are a spatial mosaic of local adaptation and maladaptation. So these are caused by natural selection, gene flow, and founder effects that drive variation and local adaptation across landscapes. So using large patches composed of many plant individuals, Nosil and his researchers mapped the occurrence of local populations of stick bugs throughout host plants.
Each ecomorph was found to be more populous on the host plant that it camouflages best to, suggesting natural selection to match the host plant. Because of the independent nature of each plant's population, stick bugs also have a short spatial range of dispersal and gene flow. Unsurprisingly, a negative correlation between the proportion of maladapted individuals in a patch and population size was found. Cooler still was that more traditional ecological factors, including habitat patch size and host plant species identity, when modeled, showed the same sort of impact. All these decreased population size and increased the likelihood of extinction.
But what is the driver of this decreased population size? Nosil and his group artificially induced maladaptation by purposely matching the wrong ecotype to each host plant in bird and bird exclusion environments. They found that bird predation both reduces the density and richness of Timema Cristinae, or the stick bugs, and arthropods cohabiting with them, because birds attracted to maladapted populations eat and or scare them away.
Nosil's group also explored another type of morph that is dark gray, called melanistic morphs, that camouflages to the stem or fruit of its host plant. Using genomic analysis data, Nosil's group conducted a principal component analysis, or PCA, of the mixed populations of melanistic morphs, green morphs, and striped morphs, and found 3 distinct clusters of genotypic variants. Figure 1b shows that these genotypic clusters almost perfectly associate with color morph, 2 green clusters and 1 melanistic cluster. Figure 1c defined 6 PCA clusters, corresponding to diploid combinations of melanistic, green, and striped. Figure 1d indicates that there are 2 main chromosomal variants, melanistic and green, leading to 3 main karyotypes.
After having identified genetic clusters, Nosil ran statistical tests to determine the mechanisms of balancing selection on the current generation and selection in the recent or distant past. To assess the current generation, Nosil tested for deviations from Hardy-Weinberg equilibrium for the 2 main melanistic and green chromosomal variants, finding that heterokaryotypes were in excess compared to their expected frequency. This is suggestive of heterokaryotype advantage selection. Color polymorphism was further found to not be recent, involving a large genomic region under balancing selection. In Figure 5c, Nosil found positive values of Hegemus D, which can arise if mutations accumulated independently among polymorphic variants, expected of balancing selection acting over extended periods of time. In Figure 5b, Nosil also found increased values of linkage disequilibrium, or LD. LD is expected to decay or build up over tens of thousands of generations by recombination, gene flow, or genetic drift. However, here it is shown to be high, indicating that multi-locus balancing selection acted consistently in populations. So what did we learn today? Nosil's work on stick bugs demonstrates the importance of Crypsis, because not only does Crypsis help stick bugs to escape bird predation, but Crypsis influences the community structure and population demography of those around it. And this illustrates the influence of ongoing evolution in our world.